Okay, so here we're going to look at chapter 11, which is DNA technology. The DNA technology is changing the world. Scientists are able to manipulate DNA today. And in an earlier chapter, we looked at the general structure of DNA, which is shown here, as you can see. Scientists are able to copy DNA, sequence it. Many of you may have actually had your DNA sequenced. If you've ever done something like 23andMe or Ancestry DNA, scientists are able to switch it on and off. They're able to probe for different types of pieces, as well as transfer it between different types of species. Now, manipulating DNA for practical purposes is called DNA technology. Applications of DNA technology include learning about life's history, relieving human suffering, such as genetic testing and genetic treatments, protecting the environment, as well as also enforcing law. DNA technology makes use of DNA. This chapter will describe some of the techniques and promise of DNA technology. DNA technology tools, using DNA technology to copy cells and organisms, and using DNA technology in medicine. If we look at DNA technology tools, we'll talk about recombinant DNA, DNA sequencing, polymerase chain reaction, or what's referred to as PCR, and DNA profiling. Using DNA technology to copy cells and organisms, this would be what we refer to as stem cell research or cloning. Okay, and finally, using DNA technology in medicine, this would be using DNA probes in pre-implantation genetic diagnosis, as well as genetic testing and gene therapy. So DNA technology can be applied to genes and genomes. The genome of an organism can be manipulated, and changing the organism's genes changes the proteins that it expresses. And here in this picture, we kind of see the whole central dogma of biology again, where you have your DNA located in the nucleus, and that will undergo a process referred to as transcription, where you will make a copy of the gene referred to as an RNA, or specifically a messenger RNA. And then that will leave the nucleus and go to a ribosome, where it will undergo a process referred to as translation, and we synthesize a functional protein. And again, these are processes that we've already talked about. So DNA can be expressed in all organisms. Here we see sea anemones have genes that encode for glowing proteins. DNA from a sea anemone was introduced into the genome of this fish. The fish is expressing the glowing protein. The fish is a transgenic organism. This is because it now contains in its genome recombinant DNA, which is genetic material that was introduced from another source. Transgenic organisms contain DNA from multiple sources. Transgenic organisms have multiple useful applications, such as manufacturing, pharmaceuticals, engineering hardy crops, and testing human genetic diseases in mice. We have oftentimes seen, for example, in areas where it's very difficult to grow wheat, they have been able to genetically modify it so that it can go in pretty arid environments. So how do scientists create transgenic organisms? There are several different ways to introduce recombinant DNA into an organism, depending on which transgenic organism scientists are making. So scientists will use source DNA to create a transgenic organism. First, scientists will acquire the DNA that they will insert into the new organism. To make transgenic bacteria, that express eukaryotic genes, it is necessary to use cDNA, which does not contain introns. And that's going to be the overall definition for cDNA. So the source DNA contains the gene that will be expressed in the transgenic organism. Scientists will then use plasmids to create transgenic organisms. They obtain a small plasmid, which is a transferable piece of DNA. It is oftentimes a circular piece of DNA, and it is a double-stranded piece of DNA. Plasmids are able to replicate themselves inside a host cell and be used for expressing the genes that they carry. Scientists will then use restriction enzymes to create transgenic organisms. These restriction enzymes will cut the plasmid and the source DNA, creating what's referred to as sticky ends. So here we have a segment of the DNA that we're going to insert, and you can see it's been cut, creating what we refer to as a sticky end, which is this little overhang. On the plasmid, you also use the same enzyme to cut the same sequence, and it will create also a sticky end. And what happens then is this end will base pair up with this end. You can see AATCC is going to pair up with TTAA. Okay. And same thing over here. And this will then insert the new segment of DNA into the plasmid. Okay. So scientists can also use bacteria to create transgenic plants. Here we have a recombinant T1 plasmid, which will combine with a plant chromosomes when the cells are infected by genetically modified bacteria. From the left, the first one is recombinant T1 plasmid, chromosomes, and herbicide-resistant genes inside an oval-shaped structure. The structure upon the infection gives a square-shaped, unaltered tobacco cell, inside which there resides a cytoplasm and mitochondria. It is connected to another cell, the transgenic tobacco cell, where the herbicide-resistant gene is present, which upon a cell division will result in a number of cells that now have that same transgenic herbicide-resistant gene. The repeated cell division results in the development of a plant. All the processes here are shown by the arrow. So you have your recombinant T1 plasmid, then you have the infection with the bacteria, then you have the herbicide resistance gene that has been integrated. This leads to cell division, and then eventually the production of the plant. And this plant is now resistant to the herbicide. So scientists can also use viruses to create transgenic animals. Scientists will use recombinant DNA, which will combine with the chromosomes when early embryonic cells are infected by genetically modified viruses. When cells divide, as the animal develops, they pass along recombinant DNA to the entire animal. Now, in order to test a lot of this and to make sure that you're inserting the appropriate DNA and that things have been assembled correctly, we will then use DNA sequencing. DNA sequencing reveals the order of bases. Sequencing DNA can be a useful tool for deducing protein sequences and determining evolutionary relationships among species. So when you do DNA sequences, what you'll have is a primer. You'll have your unknown DNA sequence, you'll have a batch of nucleotides, and then you'll have enzymes which will essentially synthesize this up. So DNA sequencing will start with DNA replication in a tube. Researchers put the unknown DNA sequence, primers, nucleotides, and replication enzymes together. Replication of the DNA occurs as it would in a cell, it's just that this is now occurring in a tube. The modified nucleotides will mark the end of each DNA fragment. Then the modified nucleotides are added to the mix, attached to fluorescent dyes. When the nucleotides are incorporated into the replicating strands of DNA, replication stops. So here you can see each of the solutions contains an unknown DNA sequence, replication enzymes, primers, normal nucleotides, which would include your adenine, cytosine, thymine, and guanine, and a small amount of the one type labeled terminator nucleotide, A, C, T, or G. The samples now contain DNA strands of various lengths, which can be detected by lasers. And what happens is the sequencing machines use capillary gel electrophoresis to sort the fragments by size. The DNA strands are separated by size. The shortest strands travel the farthest in the capillary. The laser detector and computer will then generate a readout of the actual DNA sequence. Now, sorting the DNA fragments by size reveals the sequence. Fluorescent dyes on the terminator nucleotide make the sequencing DNA simple. Each dye color represents to a different nucleotide base, and depending on the color that's being picked up by the laser, that indicates whether it's a T, a G, an A, or a C within the sequence. DNA sequence shows how different organisms are unique. Most human DNA is non-coding, which means it actually does not code for a protein. And in fact, it's actually involved in a lot of regulation. And only about 1.5% of human DNA actually encodes for actual proteins. So here we can see an illustration that shows DNA sequences of different organisms like human, chimpanzee, orangutan, a mouse, and a zebrafish. All the organisms are characterized with different graphs. The information portrayed by the vertical axis for each of the graphs is a percent of identical nucleotides compared to the human gene, values that will range from 0 to 100. While the horizontal axis portrays the beginning of the gene. For a zebrafish, the graph plots a cardiograph which oscillates between the mean line equal to 50. Major portion of the cardiograph rests below the mean line. For the mouse, the graph also plots a cardiograph which oscillates between the mean line equals 50. And if you look at the orangutan, the graph plots a cardiograph which oscillates about the line equal to 100. For the chimpanzee, it is also a line that equals 100. Okay. So you can see as far as difference, we are much more similar with this DNA sequence to chimpanzees and orangutans than we are for, say, mouse and a zebrafish here. Now, another important tool that's used in DNA technology is a process called PCR, and I have included a supplemental video also on this process. PCR stands for polymerase chain reaction, and essentially it is DNA replication inside a test tube. It is a way to take genes and amplify them so that you can make copies of it. So what if investigators only have a tiny amount of DNA to examine and they need to make more of it? Scientists are able to do that by replicating DNA using polymerase chain reaction. PCR will unwind the double helix using heat. First, the DNA polymerase, the nucleotides, and the primers are then combined with the target sequence of DNA. The mixture is heated, causing the DNA strands to separate. Here you can see you have in the tube DNA polymerase, nucleotides, primers, and target DNA sequence are all combined. You then heat up the tube, the DNA sequence will now separate. When the mixture cools, the primers will attach to the naked DNA strands, and the DNA polymerase will then replicate those strands. So what's going to happen here is the temperature starts to decrease. The primers are going to attach to each of the separated strands, and then DNA polymerase is going to make a copy of each of those. So now instead of the one copy that you had initially, we now have 2 copies. PCR will then separate the DNA strands, use the enzymes to replicate each strand, and then repeats this process, creating millions and millions of copies. So if you go through a second round of PCR, you'll produce 4 copies now. Third round, 8 copies. Fourth round, 16. Fifth round, 32, so on and so forth. Another important technique is DNA profiling. DNA profiling allows us to detect genetic differences. Investigators will use PCR to amplify DNA from a tiny biological samples. Once they have enough DNA, they use DNA sequencing to detect genetic differences between individuals. In this illustration, we can see how DNA profiling detects genetic differences in 3 parts. The first part is represented as the extract DNA from a diploid cell from 3 separate suspects. Right here, sorry. Okay, sorry about that. My phone actually just rang. So let me start back over here. So DNA profiling detects genetic differences. Investigators can use PCR to amplify DNA from tiny biological samples. Once they have enough DNA, they then use DNA sequencing to detect genetic differences between individuals. So if we actually had 3 different, say, a crime scene, and we had 3 suspects, and we also had a sample DNA that we can compare it to, what they can do is they can look at what are referred to as STRs, or short tandem repeats. They are short repeated sequences of DNA. Okay, so if you look here in this first individual, these STRs are found in every individual. So this particular short tandem repeat is AGAT. You can see it repeats itself, AGAT. What is unique is that these short tandem repeats are going to vary in number between individuals. So here we can see in individual number 1, there's 7 of them, and in individual number 2, there's 8 of them, and in individual number 3, there's 6 of them. DNA sequencing shows how many short tandem repeats are in a person's genome. These short tandem repeat patterns reveal genetic differences. So comparing the short tandem repeats profile with an individual with those of a reference profile reveals what short tandem repeats are present in his or her DNA. So DNA profiling helps to strengthen the criminal justice system. Due to DNA profiling, the number of DNA exonerations in the US has increased in the last couple of decades, as shown in this graph here. Is it ethical to use stem cells? Shifting gears here a little bit. Stem cell research is something you may have heard of, and this really goes along the political lines within this country. Some people believe that stem cells, especially embryonic stem cells, are essentially killing a living being when they do research. And other people find the value in the research that stem cells can offer, or the potential that stem cells can offer. Stem cell research often sparks public debate over the use of human embryonic tissue in research. New techniques are being used to convert adult cells into stem cells. This would actually remove the issue of having to use embryonic stem cells. So why are stem cells so important? Stem cells are important because they are undifferentiated cells. Stem cells are special because they can develop into many different types of cells. Scientists are using stem cells to develop treatments for many different diseases and to regenerate damaged body organs, and even potentially regenerate new tissue or even new organs. Embryonic stem cells are referred to as totipotent. Embryonic stem cells are able to give rise to all cell types in the body. So whether you have an embryonic stem cell, what that means is you can convert it into a neuron, you can convert it into a muscle cell, you can convert it into a blood cell, whatever you need. Unfortunately, adult stem cells are pluripotent. Adult stem cells are only able to differentiate into a limited number of cell types. So, for example, we have what's referred to as the hematopoietic stem cell, which is found in the bone marrow. And that particular stem cell will give rise to many types of blood cells, but that's all it can give rise to is many types of blood cells. It can't be converted into a neuron at that point. We oftentimes refer to these adult stem cells as already somewhat committed stem cells. All right, cloning is another DNA technology. And what cloning does is it allows you to create an exact copy of an organism. Cloning or asexual reproduction is common among single-celled organisms such as bacteria, archaea, and many protists. Plants are something that are very easy to clone. The somatic cell nucleotransfer is used to clone animals. The first cloned animal was Dolly the sheep, who recently just died a couple of years ago. What happened was an adult sheep DNA was extracted and put into different sheep's egg cell and implanted into a surrogate mother sheep. After developing from an embryo, Dolly was born. Is human cloning ethical? Human cloning could help infertile parents have children and can be used to harvest embryonic stem cells. But there are many ethical issues that surround cloning in our own species. And I actually posted a supplemental video that looks a little bit more into the ethics of this. Okay, moving along. Another type of DNA technology is the use of DNA probes to detect specific sequences. Many medical tests and procedures use DNA technology. For example, a DNA probe can be used to test whether a patient is has inherited the cystic fibrosis gene. And what they could do is they have a probe, which is a sequence of DNA that has a fluorescent label. They can mix that with some DNA, and if they see it sort of light up on the chromosome there, then that's an indication that they potentially have that gene. DNA probes are single-stranded, not single cell, single-stranded. A DNA probe is a single-stranded sequence that is complementary to a known region of the DNA, such as the cystic fibrosis allele. The probe is labeled with a fluorescent tag, and the researcher can then detect the allele using the wavelength emitted by the probe. Genetic testing can detect existing disease. DNA probes can test for many different genetic diseases in embryos, in a child, or in an adult. So another type of thing is pre-implantation genetic diagnosis. This will actually use DNA probes to reduce the odds of having a child with a genetic disease. PGD is essentially genetic testing of the embryo. PGD test embryos created in a dish. First, a man's sperm fertilizes several of a woman's eggs in a laboratory dish. This is referred to as in vitro fertilization. Researchers then extract DNA from one of the cells of each developing embryo, amplifying it by PCR, and use DNA probes to search for harmful alleles. Essentially, PGD is used to screen out defective embryos. If an embryo lacks a genetic disease, it is a good candidate for being implanted into the uterus. There's also some ethics behind whether you should do PGD or not for selecting for particular embryos. Gene therapy. Gene therapy is something that has been explored for a few decades now. Gene therapy may someday provide new treatment options for genetic diseases by replacing, if you have a faulty gene, with a normal gene. Gene therapy can use viruses. For example, cystic fibrosis is caused by a faulty gene on chromosome 7. The result is the abnormal cystic fibrosis gene. Healthy versions of the gene are placed inside of viruses, which then the patient would inhale. The viruses would then inject their genetic material, including the healthy gene, into the lung cells. And the lung cell is now able or have the capacity to actually produce a normal cystic fibrosis gene or proteins. We can also do what's referred to as genetic editing, and some of you may have heard of this, which is referred to as CRISPR-Cas9. CRISPR-Cas9 cuts and edits specific genes with unprecedented precision. Cas9 is an enzyme that binds to guide RNA that is engineered based on specific target DNA sequences. Cas9 cuts the double-stranded DNA, which is then deleted or modified depending on the goals of the gene editing process. CRISPR-Cas9 has enormous potential for applications in multiple species. Genes have been snipped out of the mosquito genome so that the mosquitoes no longer transmit malaria. Plants could be altered to boost yield, improve disease resistance, and survive a drought. Chicken eggs could be engineered for reduced allergies in humans. Broken genes that trigger illness in humans can potentially be repaired. Cancer cells in mammals could be modified so that they are more effectively targeted and eliminated by the body's immune system. Okay. Investigating life. Weeds get a boost. The herbicide Roundup kills plants via a chemical called glyphosate. Many farmers grow crops that are genetically engineered to resist glyphosate, so it would kill the weeds but not the crops. A booster gene in the transgenic crops can be transferred to the weeds, making them now resistant to glyphosate and very difficult to now control. This is due to cross-pollination. These super weeds will reproduce more. Even when there is no glyphosate, weeds expressing the booster gene produce more seeds and grow into more plants than weeds without the booster gene. Just to give you guys a little bit of an example there of how even in nature, you can have genes that are sort of transferred from one organism to the next and can sort of create this evolutionary patterns where you can create sort of these things that are going to be more fit for the environments. Okay, that is it for this chapter.